Well, I'd like to thank the organizers for inviting me to speak here today. And before I start, I just want to make sure, for purposes of full disclosure, to say that I've received research funding from, been a consultant to, or have an equity interest in these entities, in particular Eiger Biopharmaceuticals, whose drug I'll be speaking about. And before I get started, Michael asked me to say 1 or 2 minutes about my pathway to virology. And I realized this morning that actually it started here, in part, because the last time I was in this room, I was taking biology in one of these seats. It looked like a bigger room then, but I then went over to UCSF to do virology for my PhD. And then, of course, came down to the dark side at Stanford, which is a great place to do science. And I originally got into virology because I was interested in gene therapy and realized that a lot of the body's biochemistry takes place in the liver. And hepatitis viruses have figured out how to get their genes efficiently delivered and expressed in the liver, and that might be a good way to just hijack off of them to get genes delivered for gene therapy. But I quickly became interested actually in the viruses themselves and the cool biology, some of the interesting challenges that one can study. I also realized that you can do a lot of replicates, controls, and experiments in a short time, which is very commensurate with the lifetime of a graduate student, also a postdoc. And we now study all the hepatitis viruses, even respiratory viruses, influenza, rhinovirus, and are particularly interested in the molecular virology and trying to translate that into new therapies. And it's a lot of fun, and there are also sometimes some good practical consequences. And although it's not related to virology, I think my most important part of my job, I feel, is mentoring. And so I'm particularly happy and thrilled that I could share this podium with Hadass, who is one of several postdocs that Gilead has plucked out of the lab. And it's really great to be here today. So I thought I'd share with you one example of this approach of how we like to take the learnings from molecular virology into maybe a practical clinical therapy. And we'll choose an example from work with hepatitis delta virus and give you a brief overview of the delta life cycle, the role of the large delta antigen, its prenylation, the consequences for the viral life cycle, and how we've been translating that knowledge into the clinic. So hepatitis delta virus is the worst form of human viral hepatitis. It's found all over the world. The only countries that don't report delta are those that either don't test or their tests don't work. Roughly around 5% of the chronically infected B population is co-infected with delta, although that percentage varies in different parts of the world. So around 15 million people worldwide. And the reason why it's in that hepatitis B population really is found in the structure of the two virions. So hepatitis delta virus has one single-stranded circular RNA genome. It's small, about 1.7 kb, encodes for one protein, the delta antigen. And these are surrounded by a lipid envelope in which are embedded surface antigen proteins. But delta doesn't encode its own antigen proteins, it steals them from hepatitis B. So it's kind of like a parasite of hepatitis B. That's all it needs from B. B has a different genome, different capsid structure, different replication strategy. But that's why clinically delta infections are always found in association with hepatitis B. And the net effect is to make the B disease much worse. So more faster progression to cirrhosis, more fulminant hepatitis, worse survival. A recent study in the United States found among the US veteran population, there's a 300% increased risk of liver cancer if you have delta infection compared to monoinfection with B alone. So it's a bad actor, and if we could remove it, it would really help a lot of patients. Unfortunately, our current therapies don't work very well for delta. The only molecule that's had any kind of efficacy to date is interferon alpha. And even here, maybe at best a quarter of patients will respond. Among those who respond, they have to take this drug, which is injected for at least a year, if not more, high doses. And oftentimes, unfortunately, it will relapse. We don't have those SVR rates that Hadass was talking about. And even in those who do respond, it takes a long time to respond. Often, it'll take a month to even have the log decline in viral load. have been studies looking at nucleosides for hepatitis B, and those don't work, and they shouldn't really be expected to work because these molecules are very good at dropping HBV DNA in the serum, but they do nothing to the HBV surface antigen, and that's all that delta needs. And so it's not surprising that when we've done clinical trials of interferon with these HBV nucleosides, that there's no benefit at all to them. And really nothing else works. There's a long laundry list, which I've omitted here for time's sake. But I think that hopefully that's beginning to change. And this is a brief schematic of the delta life cycle, illustrating the fact that upon infection of its target cell, the hepatocyte, there's an uncoating event. The genome gets to the nucleus where all replication occurs, RNA to RNA intermediates. The newly replicated genomes acquire the delta antigen and their envelope proteins at the level of the ER where they bud out through the normal secretory pathway. All that replication that's happening in the nucleus takes place by a so-called double rolling circle model of replication, where the incoming circular template is used by polymerase activity, which goes around and around, reeling off long linear multimers of RNA of opposite polarity. These encode a ribozyme sequence, which then leads to self-processing to linear monomers, which can then recircularize to make a circular template of opposite RNA polarity and regenerate eventually the same type of RNA that comes in with the viral genome. Now, during that replication process, there's also a critical RNA editing event that occurs, and it occurs at a strategic position of the genome, such that the consequence is on the coding strand for the delta antigen, there's an A that switches to a G. And that's at a strategic position and the stop codon of the delta antigen. So that the delta antigen that's made off of this genome is the so-called small delta antigen. After this editing event occurs, translation proceeds to the next downstream stop codon. And so it adds on an extra 19 amino acids and makes the large delta antigen. So what? Well, it turns out this changes completely the function of the protein. The small delta antigen is required for replication. The large form is a potent transdominant inhibitor. And only the large form can promote packaging with the surface antigen envelope proteins. And the reason for that lies in the very end of that extra extension. At the extreme C terminus of the protein, there's a so-called CAX box, a cysteine with 3 other amino acids, and that specifies, that's a recognition sequence for the specific lipid modification by prenolipids onto this CAX box cysteine. There are 2 types of prenolipids in our body, farnesyl, geranyl, geranyl. The 15-carbon farnesyl is what's added on to the delta antigen CAX box. Both of these are originally derived from mevalonic acid. And it turns out that lipid modification is essential to make a virus particle. And we know that from molecular genetics. If we mutate that CAX box to cysteine, we abrogate the ability to be prenylated, and it completely abrogates the ability of virus particles to be made. So we can break the life cycle at the stage of assembly and release. This isn't a practical way to treat a patient, because we'd have to go into their liver, mutate all their genomes. But if we had a drug that could do the same thing, that could pharmacologically prevent that prenylation, it could be a practical therapy. And that's really an hypothesis that we've been pursuing for quite a while, trying to pharmacologically inhibit the prenylation of the large delta antigen such that we'd functionally achieve the same thing we do by molecular genetics. And the strategy we've taken is to target the last enzyme in prenyl protein synthesis. That's the so-called farnesyl transferase in the case of delta antigen that adds fully formed farnesyl onto the CAX box cysteine. And by targeting this enzyme, hopefully achieve the same functional result. And it turns out we've shown in a variety of increasing complex systems that this actually works. First, the simplest model of delta virus particle assembly, so-called virus-like particles, cell culture systems which make fully infectious virions, and in delta animal models of delta viremia. In all these systems, if you prevent the farnesylation of delta antigen, you can completely abrogate the delta virus life cycle at the stage of assembly. Here's just some example from one transgenic model where these are cohorts of mice that are either treated with one or another farnesyl transferase inhibitor versus vehicle control. And you look in the serum of these mice and there's no delta virus particles that are made. And this occurs quite rapidly compared to vehicle control. Within a week of treatment in these mice, no delta viremia is detected. More recently, we've used also a humanized mouse model for delta viremia, and there's a variety of these mice where you can put human liver into a mouse. There's really 2 essential elements. They need to be immune deficient, so they'll accept the human cells, in this case no B, T, or NK cells, and you have to do something to damage the endogenous mouse liver to create some room for the human hepatocytes to engraft. And this model, which our colleagues imported from Japan, actually, there's a herpes thymidine kinase gene that's expressed just in the mouse liver. So you give them one dose of ganciclovir and cause liver damage just to their liver, but there's no ongoing liver damage. And this turns out to be, I think, a really important part of this model. And the majority of the liver can be engrafted with human hepatocytes. And then you can take these mice and inoculate them with natural inoculums from patients, such as some HBV inoculums, achieve very high level of viremia. And then, as happens clinically, you can superinfect these mice or inoculate them now with Delta, and they'll also achieve very high levels of Delta viremia. Then you can treat them, and this is some experiment with a farnesyl transferase inhibitor, again, and really change the course of infection within days of a mouse treated with farnesyl transferase inhibitor or not. So there's some attractive features to using prenylation inhibition as an antiviral therapy. In particular, we're targeting a host enzyme upon which the virus depends. And this is a little different than direct-acting antivirals. And one predicted consequence is there might be a higher barrier to the development of resistance, because now the genetic locus that we're targeting is no longer under control of the virus. What was also nice is that there are actually orally available compounds to use for clinical trials, and this is because fortuitously there's another protein called RAS, which is modified by the same farnesyl lipid, and for that reason, not because of Delta, a lot of companies developed some potent inhibitors of farnesyl transferase. These pretty much failed for their original indication, which was oncology, and for both solid and liquid tumors, and probably is because cancer is more complicated than just RAS farnesylation, but these are exactly the molecules we'd like to use for an infectious disease like Delta. They're surprisingly well tolerated, and although Delta is our first prototype for this, there are other viruses that look like they might use prenylation, and so this could have broader implications as well. And so what we did is to then get access to one of the most potent of these farnesyl transferase inhibitors, and more importantly, one that had a lot of clinical experience before. So lonafarnib was originally developed by Schering, then Merck, and over 1,500 patients have been exposed to this drug in various oncology trials up through phase 3. The main dose-limiting toxicity was GI. And importantly, some patients have been treated for over 4 months and tolerating this. And so we've done some initial clinical studies, both at colleagues at the NIH and at the University of Ankara in Turkey, where Delta is endemic. And this is the study design for the first proof of concept phase 2A double-blind placebo-controlled study of lonafarnib done at the NIH. 2 dosing groups, a so-called low dose 100 milligram BID and a higher dose 200 milligram BID. In both groups, there were 6 patients on drug, 2 on placebo. The 2 placebo patients were offered open label enrollment in the high dosing group from the low dosing group. Short treatment duration, really designed to ask the question, can we drop viral load on treatment, and hopefully when we stop treatment, will the virus come back up, really demonstrating an in vivo effect, and is there a dose response? And the answer to both questions is yes. So here's the data. In green are the placebos. No change in viral load as expected, because Delta never goes away on its own. In blue are the low dose treated patients, about a 3/4 log reduction, and in red are the higher dose treated patients, about 1.5 log reduction. And importantly, when we take away the drug, the virus does come back. So definite in vivo antiviral effect. Even though these are small numbers, because again, there's no change in placebos, both drops are statistically significant. And importantly, there's this beautiful dose response, R-squared 0.82, between the concentration of drug that we achieve in the serum and the mean viral load drop. So in blue are the low dose patients, in red the high dose patients, here's the placebo patients. And there's no inherent limitation to this curve. So the more drug we get in, the better the antiviral efficacy. And just to put that into context, I mentioned the trials of interferon with an HBV nucleoside analog. So the HIDA2 is the largest of these trials to date. And in that trial, after 4 weeks of therapy, only a third of the patients had a log or more reduction in their viral RNA. And even after a year of treatment, only around 79% had that. Whereas in this NIH study, at the low dose group at 4 weeks had comparable percent of patients who achieved that, and at the higher dosing group, 100% of the patients achieved what couldn't even be achieved after a year of therapy with the interferon. So this was the first study in patients, and clearly demonstrated a dose dependence. Importantly, no resistance was identified. We sequenced all patients' isolates, and this really confirms the hypothesis that the host targeting mechanism, in this case, is indeed associated with a higher barrier to development of resistance. Generally safe and well tolerated. At the low dose 100 milligram group, dosing group, the investigators couldn't tell who was on drug, who was on placebo. At the higher dosing group, you could easily tell they had the expected GI side effects, nausea, diarrhea, some weight loss. But the most promising results generated to date, but we wondered, could we do better? And the reason, since lonafarnib is actually metabolized by CYP3A4, cytochrome P450 enzyme in the liver, if we could inhibit its metabolism, we could increase the post-absorption drug level and maybe even give, though, less drug at the exposure at the level of the GI tract. And so we tested this hypothesis using ritonavir, which is a known inhibitor of CYP3A4, and then asked, could we then achieve higher drug levels in this fashion? And so the so-called lower 1 study with lonafarnib with and without ritonavir, a group of patients was given this low dose, 100 milligram lonafarnib, but now with a single daily dose of ritonavir, or with standard pegylated interferon. Initially for 1 month it was well tolerated, so continued for a second month. And here's the data with the lonafarnib and ritonavir. Again, at 1 month, where before we saw a 3/4 log reduction, now we have a 2 log reduction, and at 2 months, a 3 log reduction. One of these patients actually went below the limit of quantitation at 1 month and undetectable after 2 months. And also really excitingly, along with the drop in viral load, when we looked at the ALT, these also normalized on therapy. So this is the itis and hepatitis, the inflammation, and so this was really nice to see. Here's proof that actually we were achieving what we were hoped to achieve. This is drug levels in patients. Again, at the low dose, 100 milligrams monotherapy dose, when you give us a boost of ritonavir, we have 4 to 5 fold increase in drug level in the patients. And here's the results with lonafarnib and pegylated interferon. Again, at 4 weeks, a 2 log reduction. At 2 months, about a 3 log reduction. And also improvement of ALT along with these drops in viral loads. This kind of just puts it all together. In green, again, the data I just showed you with lonafarnib and pegylated interferon. In red, lonafarnib with ritonavir. And overlaying that, the results that are seen with, again, the so-called gold standard from the HITted-2 trial, the pegylated interferon. And again, this trial was done for a year, so this is probably a better way to look at it. After a year of treatment with pegylated interferon with or without tenofovir, the mean viral load was between 2.5 to 3 log reduction, whereas now at either of these lonafarnib-based regimens, at 8 weeks, there's over 3 log reduction. And it's a much faster drop in viral kinetics. So this is a summary kind of comparing apples to apples. Here's the NIH data. Again, 3/4 log reduction at 100 milligrams, 1.5 log at 200 milligram dosing. In Ankara, same dosing regimen, achieves same results. That's always good to see. A little higher dose of lonafarnib has a better antiviral response, but more GI side effects. And so really, we're most interested now in focusing on this low dose, 100 milligram with the ritonavir boosting, which had the best antiviral response and was better tolerated than either of these higher dose monotherapy dosing regimens. And the possibility to combine with interferon is also possible, although we'd really like to finish exploring what we can do with an all-oral regimen. So the take-home messages I'd like to leave you with is, of course, Delta is a fascinating collection of biology. It's the worst form of human viral hepatitis. Prenylation is key to both assembly and a potential new antiviral target. There's a compelling preclinical rationale for prenylation inhibition-based antiviral therapy using farnesyltransferase inhibitors. And this is an example of what I'd like to call small molecule antiviral therapy targeting a specific host intracellular function, or SMART shift. It has many attractive features, especially when we think with the problem of resistance and controlling the barriers to resistance. These are the most exciting clinical results to date, but there's a lot more to come, so definitely stay tuned. And just want to make sure to acknowledge key people like Ping Liu, Menashe Elazar, and Rick Salazar, and Mark Winters in my lab, Gary Peltz and Toshi Nishimura at Stanford, our collaborators at the NIH, Jay Hufnagel, Theo Heller, and Chris Koh, Harel Dahari at Loyola, and in Ankara, Shihan Yerdaydin and his team. So thank you very much.
Do you think hepatitis D is a virus you can eradicate? Is there a kind of long-lived reservoir that you can get rid of?
Hepatitis D?
D,
Yeah, I thought you said C, and actually it's the same answer, because I think both C and D, they're both RNA viruses. There's no DNA intermediate, no DNA archiving, and so I think both of them can be eradicated.
Well, that circular RNA could be long-lived as well.
And this one is, and there's very high levels in the cell, but...
It turns over.
I'm sorry?
It does turn over.
Yes.
Because the slope was fairly slow.
Yeah, so I didn't have, for purpose of time, didn't have time to put it in here, but that's actually what Harel works on, is the viral kinetics. So as we see with hepatitis C, there is clear biphasic kinetics. And actually one thing that's very interesting from that analysis, and typically there the first phase is much faster, and then there's a slower second phase, which is involved with actually clearing out of the infected cells. So we can predict how long we need to treat, first of all. And the other interesting thing is that the onset of that first phase is so much faster than we've seen, let's say, with interferon or anything else. And the only other example where we've seen that kind of kinetics is with the NS5A inhibitors in hepatitis C. And there it's explained because of having a dual mechanism of action. And I think that's actually what was happening here probably as well, because when we use a farnesyl transferase inhibitor to prevent the large delta antigen from exiting the cell, of course, and it breaks that target's assembly. But then if it's not going outside of the cell, it can accumulate within the cell. And we know that the large form is a potent transdominant inhibitor of RNA replication. So that could be the second mechanism underlying that.
Thank you.
So in principle, you would imagine that this would not generate resistance, but is any evidence that this is the case?
Yeah, so in principle, that's kind of one of the theoretical attractive features, but actually now we have empiric data. So every patient in these trials has been sequenced at every time point, and we don't see any evidence whatsoever of resistance. And that's really nice because obviously when you develop an antiviral, you want to push to the maximum tolerated dose first, both for efficacy and also because you don't know about resistance. This is a monotherapy, actually. But now that we don't see that, we've been able to come down a lot and use much lower doses and not have to worry about the resistance. So that's very exciting, and we'll share that with you next time.
Hi, I'm just wondering the extent to which you've looked at this approach to look at other pathogens that may rely on prenylation by the host cell.
Yes, of course. So there actually are a variety of other pathogens that likely use prenylation in their life cycle, and we're very interested in actively, and there is, yes, so it does work against other viruses too, and other pathogens. This is an attractive clinical indication because we have all the preclinical rationale already. It's a huge unmet medical need. It's an orphan disease, so there's a lot of components that make it easier to quickly do those studies. But for sure, I think this can be an example of a broad spectrum antiviral therapy because of the host targeting mechanism that's used by. Whenever there's a successful solution to some problem biology by one virus, it's often used by others. Yeah, I'm sorry. By kinetics, I meant viral kinetics just based on viral load drops in serum.
Great presentation. Can you specify a little bit more about what the GI effects are, and can you incorporate some kind of symptomatic treatment into the therapeutic?
Yeah, that's a great question. So the reason I wanted to choose this particular molecule is it has GI side effects, which is a common class effect to this class of drugs, but unlike other members of this class, doesn't have other side effects. So other farnesyl transferase inhibitors have additional side effects. The main ones are some nausea, diarrhea, some anorexia, and as you suggest, there are ways to symptomatically treat those, and actually in the oncology trials, they could dramatically improve the tolerability of the drugs by using symptomatic medications, and that is also being incorporated. We've found that also as well for the antiviral indications. I would say they're less of a problem as now we're looking at much lower doses. But yes, that's an important, simple thing to do, which makes a big difference.